TCMVET Baituxiao benefits from a mixture of various herbs and mushrooms, and today we introduce the scientific literature on some of the ingredients for your reference. Hediotis diffusa. A total of 58 different antineoplastic active compounds isolated from Hediotis diffusa were discussed in this review, including iridoids, flavonoids, flavonol glycosides, anthraquinolones, phenolic acids and their derivatives, sterols, and volatile oils kind. Their anti-tumor activities include inhibition of tumor proliferation, induction of tumor cell apoptosis and tumor angiogenesis, modulation of host immune responses, anti-inflammatory and antioxidative properties, and protective autophagy. Furthermore, we provide up-to-date and systematic evidence for the anti-tumor activity and possible underlying molecular mechanisms of Hediotis diffusa. In traditional Chinese medicine, Hediotis diffusa is known for its bitter, sweet and cold properties. It is used to treat clear heat, resolve toxins and quicken blood. It has been used to treat a variety of ailments such as esophagitis, bronchitis, dysentery, mastitis, pneumonia, appendicitis, pelvitis, and astragalus. Astragalus has been used for centuries in traditional Chinese medicine in combination with other herbs. Its root is used for healthcare purposes and has been promoted as a dietary supplement for many conditions including upper respiratory infections, allergic rhinitis, asthma, chronic fatigue, infections, allergic rhinitis, and chronic kidney disease, among others. It's also promoted to strengthen and regulate immune system. Astragalus polysaccharides have been shown to regulate the immune function by enhancing the immune organ index, promoting the proliferation of immune cells, stimulating the release of cytokines, and affecting the secretion of immunoglobulins and conduction of immune signals. In terms of tumor treatment, astragalus polysaccharides have been shown to enhance the M1 polarization of macrophages, functional maturation of dendritic cells, and T-cell mediated cancer immune responses in patients with lung cancer. Another study found that extracts of Astragalus membranicus overcome tumor immune tolerance by inhibition of tumor-programmed cell death protein ligand 1 expression. Scutellaria barbata. Scutellaria barbata has a long history of medical use in traditional Chinese medicine for removing heat and toxic material, promoting blood circulation and removing blood stasis, and inducing diuresis to reduce edema. Recent pharmacology investigations have provided evidence for its anti-cancer, bacteriostasis, antivirus, anti-inflammation, antioxidation, and immunity enhancement properties. Rehmannia glutinosa. Rehmannia glutinosa is a widely used herb for clearing heat and cooling the blood. Polysaccharides from Rehmannia glutinosa have a variety of biological activities including antioxidation, hypoglycemia, immune enhancement, hematopoiesis promotion, and anti-anxiety. Rehmannia glutinosa have a wide range of pharmacological activities including anti-tumor, immunomodulatory, neuroprotective effect, hypoglycemic activity, cardioprotective, and antioxidant activities. Angelica sinensis Dls. Angelica sinensis is a well-known Chinese herbal medicine. Its root has been used historically as hematopoietic and anti-inflammatory agents for thousands of years. Modern pharmacological studies have shown that Angelica sinensis has hematopoietic, immunomodulatory, anti-tumor, antioxidant, and hepatoprotective activities, and gastrointestinal protective and anti-inflammatory effects. Baiju, Atractylodes. Atractylodes has anti-inflammatory, antioxidant, anti-tumor, anti-cancer, immune regulation, liver protection, intestinal health, promotion, neuroprotective, and other effects. Herb Paris, Paris polyphylla. It has been traditionally used for its analgesic and hemostatic effects. According to a review published in PubMed, more than 320 chemical components have been isolated from the genus Paris, including steroidal saponins, C21 steroids, phytosterols, insect hormones, flavonoids, and other compounds. Arrays of pharmacological investigations revealed that compounds and extracts of Paris species possesses a wide spectrum of pharmacological effects such as anti-tumor, cytotoxic, antimicrobial, antifungal, hemostatic, and anti-inflammatory activities. Tonglao, the rhizome of Paris polyphylla, has been used in herbal regimes to treat parotitis, mastitis, and certain malignant tumors for thousands of years in traditional medicine. Polyphylline 1 is the main bioactive component in Paris polyphylla. Recent studies of PP1 in various types of cancers have shown that polyphylline 1 may exert a broad spectrum of anti-tumor effects, including inducing cell cycle arrest, inducing cell apoptosis, inducing autophagy, anti-angiogenesis, sensitizing tumors, and chemotherapy, and participating in modulation of inflammatory and immune response. Along with the growing research interest in polyphylline 1 as well as accumulation of experimental evidences, this review periodically summarized the recent advances in regard to polyphylline 1's anti-tumor properties in various cancers and the underlying mechanisms for future prospective research. Curcuma fiacalis. Curcuma fiacalis is used in China as health food and folk medicine for removing blood stasis, alleviating pain, and tumor therapy. According to a study published in PubMed, it has anti-inflammatory, antioxidant, and anti-tumor activities. Prunella vulgaris. Prunella vulgaris is a perennial herb native to Europe, Asia, Africa, and North America. It has been traditionally used as an anti-inflammatory, antipyretic, anti-rheumatic. According to a study published in Science Direct, it contains flavonoids, organic acids, polysaccharides, pentacyclic triterpenes, and volatile oils. It has a broad range of biological properties including anti-cancer, antioxidant, antiviral, anti-inflammatory, anti-allergic, and antibacterial activities. Red peony root. Red peony root is derived from Paeonia lactiflora and has been found to exhibit a range of pharmacological effects. Studies have shown that it has antioxidant, anti-inflammatory, anti-tumor, cardiovascular protective, and neuroprotective properties. The therapeutic effects of red peony root on the nervous system include neuroprotection, antidepressant, sedative, analgesic, and anticonvulsant effects. Suberect spatholobus stem. The vine stem of Spatholobus suberectus is used as a traditional herbal medicine in China. Chinese herbal medicines are well known as natural bioactive compounds that can be used as new medicines, and their antioxidant and anti-cancer effects have also been reported. One study found that an ethanol extract of Spatholobus suberectus stem effectively suppressed the viability and proliferation of rat C6 glioma cells through an antioxidant effect. The extract was found to regulate reactive oxygen species levels, induce mitochondrial membrane depolarization, and increase P21 protein expression. Another study characterized the major phenolic components of Spatholobus suberectus stem as homogeneous B-type procyanidine conjugates using a liquid chromatography with diode array detection (ESI) mass spectrometry method. This is the first report on the occurrence of most B-type procyanidins in this herb. Spatholobus suberectus inhibits cancer cell growth by inducing apoptosis and arresting cell cycle. Root of catclaw buttercup, Ranunculi ternati radix. In traditional Chinese medicine, it is known for its ability to resolve phlegm and dissipate nodulation, remove toxicity, and resolve swelling. It is used in the treatment of scrofula, malaria, pulmonary tuberculosis, snake and insect bites, toothache, pharyngitis, migraine, and deep-rooted boil. According to a review of its chemical constituents, pharmacology, quality control, and clinical applications, it mainly contains glycosides, organic acids, esters, sterols, flavonoids, alkaloids, and volatile oils. The pharmacological activity of ranunculi includes immune regulation, anti-tumor, anti-tuberculosis, antioxidant, antibacterial, hepatoprotective, and anti-inflammatory effects. This plant is especially effective in the treatment of cancer, tuberculosis, thyroid, and nasopharyngeal disorders. Appendiculate cremaster pseudobulb, Shansugu. Shansugu is a traditional Chinese herbal medicine known as Pseudobulbus cremaster. It has the property of relieving fever, counteracting toxicity, dissipating phlegm, and resolving masses. At present, 190 compounds have been isolated from appendiculata, including phenanthrene, dihydrophenanthrene, bibenzyl, flavonoids, terpenoids, glycosides, aromatics, and other compounds. These compounds have been reported to have a variety of pharmacological activities such as anti-tumor, antibacterial, anti-angiogenesis, antioxidation. Dandelion. In traditional Chinese medicine, dandelion is referred to as Pu Gongying and is considered to be energetically bitter, drying, and cooling. According to TCM, dandelion clears heat from the liver and has a beneficial effect on the stomach and lungs. Dandelion has been studied for its effects on digestion, inflammation modulation, and immune modulation. It also is full of potent antioxidants. In terms of its effects on tumors, studies have shown that dandelion extract can induce breast cancer cell apoptosis and exhibited anti-tumor properties. Poria cocos. Poria cocos is a fungus that grows in diverse species of pinus. Its sclerotium, called fulling, is used in traditional Chinese medicine for its diuretic, sedative, and tonic effects. It has a variety of pharmacological effects such as a reduction in swelling and inflammation and detoxification. In terms of its effects on tumors, Horia cocos has been reported to have anti-tumor properties. Its main active ingredients are triterpenes and polysaccharides, which have been reported to have pharmacological effects on anti-tumor, antibacterial, antioxidant, anti-inflammatory, immunomodulation, and liver and kidney protection. Chinese honeysuckle. Chinese honeysuckle has been shown to have pharmacological effects. In traditional Chinese medicine, honeysuckle is used for detoxification, anti-inflammatory, anti-lipid, anti-endotoxin, and antioxidant properties. It is believed to help cure colds, fever, inflammation, swelling, boils, sores, and viral and bacterial infections. Gardenia fructus. Gardenia fructus is one of the most commonly used Chinese herbs in traditional Chinese medicine and has been used for over 1,000 years. It is usually prescribed for treatment of brain aging, vascular aging, bone and joint aging, and other age-related diseases. It has been demonstrated that several effective compounds of gardenia have neuroprotective or related activities which are involved in senile disease treatment. These bioactivities include the mitochondrial dysfunction, antioxidative activity, apoptosis regulation, and an anti-inflammatory activity, which related to multiple signaling pathways such as the nuclear factor pathway, AMP-activated protein kinase signaling pathway, and the mitogen-activated protein kinase pathway. To lay the ground for fully elucidating the potential mechanisms of gardenia in treating age-related pathologies, we summarized the available research conducted in the last 15 years, which have been studied in vivo and vitro. Fritillaria thunbergi. Bulbous Fritillaria thunbergi has been widely used as an antitussive herb for thousands of years in China. It has been used to clear heat, resolve phlegm, relieve cough, remove toxicity, and disperse abscesses and nodules. 13 pharmacological effects of Fritillaria thunbergi were identified, including anti-cancer, tracheobronchial relaxation, antitussive, expectorant, antimuscarinic, anti-inflammation, anti-thyroid, regulation of blood rheology, anti-ulcer, anti-diarrhea, pain suppression, antioxidation, and neuroprotection. Sparganium rhizoma. Sparganium rhizome, also known as Spargani rhizoma, is a traditional Chinese medicine that has been used for hundreds of years. It has been used for eliminating blood stasis, promoting the flow of qi, removing the retention of undigested food, and relieving pain. As an important traditional medicine, Sparganium rhizoma has been demonstrated by modern pharmacological researchers to have significant bioactivities, especially on anti-tumor, antithrombotic, and estrogen antagonistic activities. Arnebia euchroma. Arnebia euchroma is native to Inner Mongolia and northwest of China. It holds promising potentials for anti-tumor effects via multiple target mechanisms. Shaikonins, alkanin, and their derivatives are the main ingredients of Arnebia euchroma and have shown anti-tumor activity in apoptosis, necroptosis, and immunogenic cell death. Sichuan lovage. Sichuan lovage rhizome. It has the effects of promoting qi and blood circulation, dispelling wind, and relieving pain. Because it has a reputation for promoting blood circulation and removing blood stasis in China for thousands of years. In addition, it also has antioxidant, neuroprotective, anti-inflammatory, antibacterial, anti-proliferative, and pro-apoptotic activities. Tianqi ginseng. Panax noto ginseng. More than 200 chemical components were isolated from Panax noto ginseng, including saponins, polysaccharides, dianthus, amino acids, flavonoids, phytosterols, cyclic peptides, sugars, fatty acids, volatile oils, and trace elements. It has multiple effects: hemostatic and wound healing activity, antioxidant activity, anti-inflammatory activity, hypoglycemic and anti-hyperlipidemic activity, anticoagulant activity, neuroprotective effect, hepatoprotective effect, renal protective effect, anti-tumor activity, antivascular senescence, therapeutic potential and cellular mechanisms of Panax noto ginseng on prevention of aging. Eclipta prostrata, also known as Eclipta, is a traditional herbal medicine that has long been used in Asia and South America for the therapy of hemorrhagic diseases. Examples include hemoptysis, hematemesis, hematuria, epistaxis, and uterine bleeding. Skin diseases, respiratory disorders, coronary heart disease, hair loss, vitiligo, snake bite, and those caused by deficiency of liver and kidney. Various phytochemical classes have been identified and isolated from the plant, covering triterpenes, flavonoids, triopenes, steroids, and others. The isolated crude extracts and individual compounds have been reported to exhibit promising pharmacological properties such as hepatoprotective, osteoprotective, cytotoxic, hypoglycemic, anti-inflammatory, antimicrobial, hypolipidemic, promoting hair growth, rejuvenative, and neuroprotective effects. Chinese licorice. Chinese licorice root is derived from the root of Glycyrrhiza urolensis or Glycyrrhiza glabra. It is a common traditional Chinese herbal medicine that has several pharmacological effects such as anti-inflammation, detoxification, antibacterial, antitussive, and immunomodulatory effects, especially in respiratory diseases. Chinese sage, Salvia chinensis. Salvia chinensis, also known as Chinese sage, has been widely studied for its pharmacological properties. In China, about 40 salvia species have been used as medicinal plants for treatments of various diseases, specifically hepatic and renal diseases, and those of cardiovascular and immune system. Has been widely studied for its anti-cancer properties in a variety of cancers. Another study found that a polyphenol-rich extract of salvia exhibited anti-cancer activity in different cancer cell lines and induced cell cycle arrest, apoptosis, loss of mitochondrial membrane potential in pancreatic cancer cells. Schisandra, 5-flavor berry. One study found that Schisandra chinensis has high potential for beneficial health effects. Its constituents exert anti-cancer effects through the induction of cell cycle arrest and apoptosis, as well as inhibition of invasion and metastasis in cancer cell lines and experimental animals. Asian ginseng. Asian ginseng has been shown to have a wide assortment of therapeutic applications for animals, including the treatment of irregular weight loss due to cancer. It induces cell differentiation, reduces the effects of chemical carcinogens, mitigates inflammatory carcinogenesis, induces apoptosis, inhibits proliferation. Ganoderma lucidum. Ganoderma lucidum has been shown to have anti-tumor effects mainly because of two important components in the mushroom, triterpenes and polysaccharides. Studies have shown that reishi can inhibit tumor growth by 88% or more, depending on the case. In some animals involved in the study, the tumor disappeared completely. Even when reishi does not completely eliminate tumors, it still prevents them from growing. It is believed to modulate many components of the immune system such as antigen-presenting cells, natural killer cells, T and B lymphocytes. Turkey tail mushroom. Turkey tail mushroom contains beta-glucan polysaccharides which are thought to stimulate natural killer cells, boosting your dog's immunity and healing abilities. A study published in Evidence-Based Complementary Medicine looked at the effectiveness of turkey tail mushrooms in Dome's immunocarcinoma. Turkey tail mushroom. Turkey tail mushroom contains beta-glucan polysaccharides which are thought to stimulate natural killer cells, boosting your dog's immunity and healing abilities. A study published in Evidence-Based Complementary Medicine looked at the effectiveness of turkey tail mushrooms in Dome's hemangiosarcoma is a form of vascular cancer that spreads quickly. It's a highly aggressive condition that's mostly found in dogs' spleen.